Well, hello everyone. Thanks for joining our webinar. I am Kutay Ozturk, Extension Potato Pathologist with the University of Maine. And here today I am introducing Shan, research scientist from University of Wisconsin-Madison, to talk about her research on soil fumigation and how does it affect soil health. So the time frame for this presentation will be 45 minutes to talk and 15 minutes for questions and answers. Feel free to ask your questions in the chat anytime, and we will address them at the end of the presentation. And there will be one pesticide credit for Maine growers. Kerry Bernard will be sharing the link to the quiz. At the end of the presentation, you will need to fill the quiz to get the pesticide credit. Thank you. I will let Shan to talk now.
I just interrupt for 1 second. It looks like the extension's website is still down, which means the online quiz won't work. But so what I'm going to do is I'm going to send an email with the quiz questions so folks can still see those and maybe jot down the answers as we go. And I'll also upload a Word document of the quiz into the chat. But if folks could still do the online quiz later today, because I'm sure the website won't be down long, that would be great. Thank you.
Thank you.
All right. Thanks everyone for coming to the talk today. And thanks Kutay, Carrie, and the University of Maine for this chance to talk about my research. I'm Shan, and I'm a researcher at the Lankau lab at the Department of Plant Pathology at the University of Wisconsin-Madison. Our lab works with Wisconsin potato growers, primarily on soil health and soil microbiome. Today, I'm going to talk about some of the research we did in the past that is related to fumigation. Hopefully, my talk can give a majority of you some good, useful information, take-home message. And I'll also take any questions. Feel free to contact me if you have any question. My email is on the bottom of this slide. Because our lab works studies primarily on soil microbiome and their functions in agroecosystems, and fumigation is the one practice that mostly targets on soil microbiome. Naturally, we became interested in this topic. We are interested in fumigation and the use of fumigation in sustainable agriculture. A couple of years ago, we reached to our growers and asked them to help us to study a research where we can investigate the ecological consequences of fumigation. They were very supportive. They offered to set up plots in their field, and they refused to take any economic compensation from us. So it was great working with them, and because of them, I was able to do some research about fumigation, and I'm able to talk about this today. All right. Fumigation. Soil fumigation is a globally employed practice. It is used on high-value crops that are sensitive to yield loss, because many of the high-value crops are subject to monoculture and the disease pressure. The largest yields of fumigation are usually in growing potatoes, tomatoes, strawberries, and carrots in the U.S. Fumigation can be applied in the fall or spring before crops are planted. This chemical is a phytotoxic, so it is recommended to wait for 2 weeks or 3 weeks before planting your crops and after fumigation. The chemical can be directly applied by injection or through your irrigation systems. Once the chemical is in the soil, the chemical will turn into gas. The gas can travel between the soil particles in the air and in the water and kill organisms it comes into contact with. Why do we fumigate? Because fumigation is very effective at controlling a wide range of pests, including parasitic nematodes, plant and fungal and bacterial pathogens, and also insects and weeds. A variety of chemicals can be used as fumigants. This includes methylbromide, which was one of the most effective chemical fumigant in the past. But the use of methylbromide was started to phase out in the year 2005 because this chemical is associated with ozone depletion. There are less viable alternatives to methylbromide at this moment, including chlorpicrine, dichloropropane, metasodium, etc. These chemicals are less effective as compared to methylbromide, and they also come with variable efficacies when treating different pests. The main justification behind fumigation is to control soil-borne diseases. The pathogens living in the soils are extremely difficult to treat, to eliminate. They live long in the soil and produce spores that live in the plant tissue and survive for a long time. They also spread easily through contaminated soil particles and water. It can spread through the dirt that is attached to farming equipment or when people walking across fields. It is difficult to predict the outbreak of those soil-borne diseases. The inoculum level of the pathogen in the soil does not necessarily translate into the disease level in the plant. The reason for that is the outbreak of those diseases need certain inoculum level in the soil, but also favorable environmental and climate conditions. For some of the soil-borne diseases like Bacillium, we have a few resistant crop varieties that are available. And our current control methods are not perfect. One control method is crop rotation. Growers can choose to incorporate some crops that are not host into the crop rotation cycle, and that sometimes would break up the host pathogen cycle, but it does not always work, especially for those pathogens that live in the long term in the soil. Growers can also choose to grow brassica cover crops in the brassica family, such as broccoli, turnip, radish, canola, rapeseed, and mustard. These cover crops can produce a compound very similar to fumigation that is toxic to the pathogens. But the reality is this method is not always applicable and is not always effective. That is why we fumigate. History of fumigation. Fumigation started a long time ago, in the year 1927 to 1928. First government stock of chlorpicrine became available for control of nematodes in banana plantation. After that, in the year 1941, methylbromide entered the market. By the mid-19th century, fumigation became a common pre-planting practice for most vegetable crops. In the next 5 decades, the use of fumigation rapidly expanded. And we mentioned earlier, in the year 2005, methylbromide started to phase out of the market. By the year 2019, in the US, nationwide, chlorpicrine is applied to more than half of the strawberry acres, and metasodium is applied to 24% of the potato acres. But fumigation is not perfect. It has problems. First, it is expensive. Fumigation is an expensive practice that costs significantly in the potato production. The price of fumigation has increased more than 50% in the past decade. On the other hand, the chemical we use in the fumigation is toxic. That's why when you apply the chemical, the application is facing very strict government regulations. The chemical used in fumigation are also broad spectrum. That means the chemicals not only target the pests in the soil, it also targets non-pathogenic microorganisms. And this is where our research interest lies, which is the potential risks of fumigation to the soil microbial community and the soil health. Sorry, I need to remove my bar a little bit. Okay. Soil is complicated. It is an ecosystem composed of air, water, minerals, organic matter, and hundreds and thousands of macro and microorganisms living in them. This complicated ecosystem provides many different functions that is related to every aspect of our daily life, including food security, food production, clean air, water, climate control, etc. The thing is, all those functions are linked with each other because majority of the soil microbial components, the microorganisms participated in most of the functions. So when we manage one function, we run the risks of influencing other soil functions. That is why managing soil-based problem is always difficult. For instance, like fumigation here, fumigation is a method we use to improve yield and provide food security to us. But it also has problems because it targets both pathogenic and non-pathogenic microorganisms, and that could cause some potential problems. Plants are not individual living organisms. There are microbes living on the roots, on the plant surface, in the soil surrounding them, in the rhizosphere, in the bulk soil. And those microbes are playing an essential role in the plant growth. And actually plants depend on them to grow healthy. The functions of the microbes in the soil and in the rhizosphere and on the plants there are many functions they can perform. Some of the microbes can produce plant hormones and directly improve plant growth. A lot of them, the majority of the microbes participate in nutrient cycling. We know that the nutrients in the soil are usually in a form that is not available for plant use. They are trapped in those organic compounds. It's difficult for a plant to just take up nitrogen, phosphorus from them. They need to be break down into small molecules and into inorganic forms such as ammonium nitrate, which plant can directly take up. So microbes are the ones who produce enzyme, break down all those organic compounds, go through nitrogen mineralization, phosphorus mineralization, and turn them into those available forms for plant use. Microbes also directly social with plant roots, and then they can take up organic compounds and then break up those compounds inside their tissue, which is also inside the plant tissue, and then give the nitrogen to the plants. A lot of the microbial community members also are direct competitors against soil pathogens. We know the concept of suppressive soil, which means there are members in the microbial community in those soils that are suppressing pathogens. When we fumigate the soil, we run the risk of taking away some of the good microbial members along with their beneficial functions. Over time, that could lead to two consequences. One is without those beneficial competitors, you leave the door wide open for the pathogens to come back even stronger in the following years. If you get the soil this year and the beneficial microbes population drops a little bit, or the community change a little bit, and then the pathogen can come back stronger the next year. And then in the next year, you would be facing this problem, a more severe pathogen problem, and then you would need to fumigate more next year, or depend on the fumigation more next year. We haven't worked for this. It is a pesticide treadmill effect. Basically, as the year goes on, you will have to fumigate more and more just to keep the disease pressure to a certain low level. Right. So the other consequences, there's another problem associated with fumigation is without those microbes, we're talking about those microbes helping plants taking up nutrients. Without the microbes participating in nutrient cycling, this nutrient transformation process could be inhibited in the soil. Without those extra nutrients, when we're not fertilizing the soil, when the microbes are providing plants with nutrients, without those microbes, plants might need more fertilizer from us. There might be a chance or there might be increased need for more heavier fertilizer with fumigation, just so that we can sustain the previous yield levels. Okay, with those concerns in our mind, we did a study a couple years ago, and we asked the question: how does fumigation affect soil functions? We look at tuber yield, tuber yield, tuber disease, and a bunch of other parameters that measure soil health. One hypothesis we had is fumigation could induce treadmill effect. The other hypothesis, fumigation could decrease or increase the need for heavier fertilization. To understand the fumigation effect on soil health, we took the approach where we compare soil functions or soil health in the fumigated and non-fumigated area within the field. In the fall of 2020, before growers fumigate their fields, we asked them to leave a small section of their field unfumigated for us. So the growers left a small strip in across their field for us. The strip is about 20 to 50 feet in width. The length vary depending on the field, or depends on what the grower wanted to do. So these strips are unfumigated. We can collect samples within those strips, and we can also collect samples outside the strip in the area where it is fumigated as a comparison. We selected 7 commercial fields across Wisconsin in 2 locations, in 2 growing regions. One is central Wisconsin sandy soils growing region. The other is northern Wisconsin loamy soil. The field size varied between among like 35 to 63 hectare. Metasodium was fumigated. Metasodium was applied in the fall through injection at a rate of 37 to 40 gallon per acre. And in the next growing season after the fall application of fumigation, we collect samples. We compare the soil functions between the fumigated and non-fumigated areas. And at the end of growing season, we collect potatoes to measure yield and disease. So here, this photo shows that the soil in the 2 growing regions we collected from are quite different. In central Wisconsin, we have this very thick and coarse sandy soils. The soils has very coarse texture and pretty much has very limited holding capacity for any nutrient or water. As a contrary, the soil in northern Wisconsin is very different. It is much more. It has a finer texture. It holds more organic matter and nutrients. And when it rains, it forms a nice texture. This photo here shows you the unfumigated strip in the field. As you can see on the left part of this photo, where the plant coverage is low, that is the unfumigated strip. But in the early growing season, we're looking at look at the plant between the fumigated and unfumigated strips, unfumigated area. There wasn't that much difference in terms of how dense the plant looked like. It was in the later growing season, towards the later part of the growing season, in some of our fields, the strips start to show. You can see plants in the strips are dying quicker or earlier than the outside the strip. So our first question is how does fumigation influence tuber yield, disease, and soil health? Our first figure shows you the yield response to fumigation in the 2 different growing regions, central Wisconsin and northern Wisconsin. Lavender represents central Wisconsin fields, and this dark mustard color represents northern Wisconsin fields. The bar with the slash in it, or with shade in it, represents fumigated area, and the bar without represents the unfumigated area. And then you can see that the potato or the tuber yield in the fumigated area is much higher than the unfumigated area for the central growing regions, for the central Wisconsin fields. But the trend is reversed in northern fields. So that suggests that the metasodium fumigation increased tuber yield in the central sand, but not in the northern Wisconsin loam soils. We look at tuber disease. We measured potato common scab with 3 different indexes: the incidence of superficial scab, the incidence of pitted scab. Basically means the frequency of the tubers getting infected. And also scab severity. Scab severity is a measurement of the incidence of scab and also the percentage of the scab coverage on the infected tuber surface. As you can see, there was no not much difference between the fumigated and unfumigated region in terms of scab disease. So we conclude that fumigation seems to not have much impact on the scab disease on the tuber. We also looked at. We also want to look at potato early die, the Verticillium infection. We don't have a good index to quantify potato early die. Instead, we cut off the tuber and look for the discoloration of the vascular ring, and use that as a representative of the infection of the Verticillium. So we found that the Verticillium infection was less in the fumigated area compared to the unfumigated area, and that pattern is consistent. Is consistent between regions. So we conclude that fumigation decreased the instance of vascular discoloration in the tuber in both regions. We also quantified the pathogen in the soil. Use qPCR. The method we use here is different than what people would get if you send your sample to a commercial lab. So those commercial labs, they usually take a scoop of soil and grow them on a petri dish and see how many colonies of the pathogen can grow out of it. What we did was we grind up the soil, extract the DNA out of it, and quantify the pathogen DNA with a probe. This method is quicker, but and the results is sometimes it's comparable. I have compared this method to the traditional plating method in the commercial lab. Sometimes you can see good correlation of those two methods. We quantified the abundance of two pathogens. One is the streptomyces, which is a causal agent for the scab disease. The other is Verticillium, which causes potato early die. We found no difference in those pathogens quantity between the fumigated and unfumigated areas. So fumigation seems to not impact the abundance of those pathogens in the soil. But one thing I want to mention here is this analysis was measured. The analysis of soil pathogen was performed in the summer during the growing season. Fumigation happened in the fall, in the previous fall. So it has been 9 months after fumigation when we measure the soil pathogen abundance. We did some parallel study in our lab, and we actually found that the pathogen level, especially the Verticillium level in the soil, actually did decrease immediately following fumigation. Like if you measure 1 week or 1 to 3 weeks following fumigation, you're likely to see a decreased pathogen load in the soil. But according to this data, they seem to bounce back after crops are planted. One thing I was interested to look for here is earlier we saw this interesting response pattern in the yield, where the yield was increased by fumigation in central Wisconsin but decreased by fumigation in northern Wisconsin. If we can find a similar pattern in the disease response, we can make some interesting link between those two responses. But unfortunately, there was no such pattern in the disease response to fumigation. That means whatever we observe in the yield response to fumigation may not be associated with the disease responses. So that means if fumigation increases disease, if fumigation decreases disease, that does not necessarily mean fumigation will increase your yield. And then we look at the soil health, the fumigation effect on soil health, which is the most important part, most interesting part of our research. The first thing we looked at was the microbial diversity. Why do we look at microbial diversity? Diversity means higher diversity means there are more different taxa, microbial taxa in your system. Diversity is usually associated with resistance, ecosystem resistance, resilience. One of my other research actually shows that sometimes diversity in the bacterial community is actually associated with the ability for the soil to suppress diseases. So we first look at the bacterial diversity. This figure here shows you that bacterial diversity was increased by fumigation in the central Wisconsin soils, but was decreased by fumigation in the northern Wisconsin soils. Clearly, this is a similar pattern with our yield response to fumigation. How about the fungal diversity? We saw a consistently lower fungal diversity with fumigation in both regions. That means metasodium is pretty consistent at suppressing fungal community. This is not surprising because the metasodium is considered primarily a fungicide. Another way to look at microbial community is to look at who's there, the composition or the makeup of the community. The way to do that is plotting. We can plot a microbial community as a single dot on a two-dimensional space, and the distance between two dots or the two communities represents the dissimilarity between the two communities. So the further away the two dots are from each other, the more different the two communities are from each other in terms of their makeup. We did that to our data, and we found some sort of a separation before the fumigated and non-fumigated microbiomes. So that suggests that fumigation shifted the microbial community composition. But the direction of this shift is not consistent among fields. For instance, like in this red field, our microbial community shift from here to here, from non-fumigated to fumigated. And for this turquoise, field, you can see the community shift from here to here. What does that mean? That means we were worried about earlier that fumigation would just wipe out everything. If fumigation wipes out everything, then all the communities is going to look similar after fumigation. But this suggests that fumigation didn't really just wipe out everything and make all the community look similar, but rather fumigation shifted each community a little bit, the composition a little bit. So we see a slightly different community after fumigation. That suggests the community is still there, and the basic structural integrity is possibly also still there. We saw the very interesting response pattern of yield to fumigation, and then that made me to think what caused those response patterns. What does the yield response to fumigation depend on? Like when would you observe a positive fumigation effect on the yield? When would you observe a negative fumigation effect on the yield? To do that, we did a bunch of regression analysis, and then I'm just going to show you the result. Here, the first result is a figure shows you the Y represent yield response to fumigation. Positive value, if Y is positive, that means your fumigation increased yield. If the value is negative, that means fumigation decreased yield. The x-axis here represents bacterial diversity response to fumigation. Same here, if the value is positive, it means fumigation increased bacterial diversity, and if it's negative, it means fumigation decreased bacterial diversity. When we plot those data on this figure, and we found that those two factors, these two yield response to fumigation and bacterial response to fumigation, they are positively correlated with each other. So it basically means when after fumigated, if your bacterial diversity in your field increased following fumigation, your yield is also likely to increase in response to fumigation, and vice versa. The other interesting thing we found here is the second figure. The Y is the same thing, yield response to fumigation, but X here represent the native soil bacterial diversity. And it means the soil bacterial diversity before fumigation was applied. If your soil's native bacterial diversity was high, like your field will be located on the right part of the map, and then you're likely to see a low yield response to fumigation. That means the fumigation wasn't doing much to improving the yield, or was sometimes even decrease the yield. And on the other hand, if your soil has a very high bacterial diversity to begin with, and if you fumigate it, you would likely see a very high. If your field has low bacterial diversity to start with, has very low bacterial diversity to start with, you're likely to see a high response of yield to fumigation. That means the fumigation is going to do lots of benefit to the yield. What is really interesting here is if you look closer to this figure, if you start with a field that has a high bacterial diversity, like a very organic, highly organic field, or like a field that is recently converted to agriculture land, still maintaining its old bacterial diversity, and you fumigated that field with high diversity, and if the bacteria in that field was damaged, that bacterial diversity was damaged by fumigation, you can push that field towards the left part of this figure. That means the yield response is going to increase as the bacterial diversity was damaged by the fumigation. So basically, once you fumigated, once you fumigate a soil with a high initial bacterial diversity, it's likely to be followed by increased dependence on the fumigation for the yield for future. And this is what we talked about earlier, the treadmill effect. So when the treadmill effect could happen, suggested by this figure, it means treadmill effect may happen where your native soil bacterial diversity is high and your soil bacterial diversity decreased following fumigation. Another aspect of soil health are the soil chemistry. We look at the microbial parts, and now another important part is the chemistry part of the soil. Overall, we did not find any fumigation effects on soil chemistry, These include the micronutrients like the NPK and a bunch of other micronutrients. And also soil pH, organic matter content, carbon, protein. None of them showed any response to fumigation. How about microbial functions in producing those nutrients? Earlier we were talking about what about fumigation could negatively damage microbial function in the nutrient cycling. We look at enzyme activity, with nitrogen decomposition, phosphorus decomposition, nitrogen mineralization. The enzymes are the ones that break down organic matter into inorganic matter, and mineralization. Nitrogen mineralization is also a process happening on the microbes or inside the microbes, where the microbes take up organic nitrogen and put out ammonium for plant use. We found that fumigation did not influence these processes. So basically, fumigation did not really affect the ability of soil microorganisms to produce nutrients. This is consistent with our earlier observation of shifting the community composition under fumigation rather than the completely suppressed community composition under fumigation. It means the community is still sustaining some of its functions, such as nutrient cycling. So for our second hypothesis that the fumigation may increase the need for heavier fertilization is not supported. Take-home message. Metasodium fumigation does not always increase yield. It increases yield when your native soil bacteria diversity is low and your bacteria diversity positively responded to fumigation. Example for this case includes soils that have been repeatedly fumigated in the past, or soil with very poor microbial functions to start with. Fumigation may lead to treadmill effects where your native soil bacteria diversity is high and the soil bacteria diversity negatively respond to fumigation. Example for this scenario includes naive fields, like the field that was natural land before but was recently converted to agriculture land, or if your field has always been organic. You may fumigation may lead to treadmill effects. And at last, fumigation does not seem to influence the nutrient availability. With that, I'll take any questions from now on.
Thank you, Shan. That was very informative. I really appreciate taking the time to talk about that. So, any questions from our attendees? You can type in the chat. You can speak if you would like. I have a couple questions then. So first of all, did you see any significant change, whether it increase or decrease, on certain members of the soil microbiome, which are known to have some reported effects on potato health?
Yeah, that's a good question, and we did see some members increase following fumigation, which is kind of surprising. We see a particular member's actinobacteria increased following metasodium fumigation. We know actinobacteria is usually involved in disease suppression. So it is interesting to see that the fumigation might actually select this particular group of beneficial organisms. It is not only metasodium. Actually, in our other studies, we have sometimes observed these kind of phenomena with like chlorpyrifos fumigation or in different fields.
Okay, thank you. The other question is, what potato varieties were grown in these regions?
So we have multiple varieties. We have Gold Rush. There's a cheaper. There's like a couple of cheapers. White potatoes, and then there was there's Burbank. And what else? And there was also a yellow potato variety. I don't have that particular in mind, but it was a variety of different varieties. There was a series of different varieties.
And did you see any variety-dependent effects? Does some varieties respond differently than the others?
Yeah, that's a good question. Like for the yield, when we see like one variety, when I have two different varieties in terms of their yield, it really mostly depends on the region. It is where it's from, not depend on what variety you're looking at. Of course, we hope to get more data in the future, and then that would include more varieties in this analysis. It's always great to have more. It's possible that some of the fine details were not captured sometimes.
Okay, well, it's a short question, but what is the next step for you?
So this fumigation strip study, gladly we talked to Gore, and Gore's like, yeah, we would like to continue doing this. So they left the strips unfumigated for us this year again. This is after a couple of years after rotation cycle. So we kept all those fields, those plots. We're gonna go back this year to measure them again. And also another thing we found is, well, we see these variations of response, yield response to fumigation, and then it's likely that there's a lot of places in Wisconsin or even outside of Wisconsin, growers are not benefited that much from fumigation. So we did so we're going to do a study where we go outside the sample a lot more fields and then see do we actually always see yield increase following fumigation? And then for that particular project, we also have economic analysis involved. So we're going to look at, are actually growers always benefiting from fumigation? If not, on what kind of soil, what kind of condition you will benefit from fumigation, and when you would not benefit from fumigation.
Yeah, and Bruce has a question. When would you recommend fumigation?
Yeah, like so the recommendation I provide for this presentation is based on this study, right? So I have to warn you that it is one single study. But what we found in this study is if your field has never been fumigated before, and when you first fumigate it, you may not see much good benefit out of it. And if you fumigate it more, you may depend on the fumigation. You will start to see good benefit, but you may depend on the fumigate more in future. But like I said, I have to stress, I have to make this point clear. This is based on one-time study. I think more empirical data is needed to give you a very solid, like great answer.
Yeah, I found it really interesting when you looked the verticillium population basically bounced back to where it was in mid-season. Yes, when it's first applied, of course, it killed most of it, but whatever is left, it was enough to restore the population of verticillium. So
Right. Yeah, that's the resistance of soil pathogen.
Yeah. Thank you so much. Yeah, are there any other questions? And you can always ask Shan through the email address she provided. We will include this webinar in our website, and feel free to access anytime or let your friends or colleagues know. I found it really informative. Thank you very much.